Okay, in this video, I'm going to explain what happens when the chromatin opens. So in the chromatin, there is a DNA, and based on the food, different foods have a different impact on what types of gene transcription factors open and which ones are enabled and which ones work. So the chromatin has to be opened, of course, for this to work. Because if it's closed, then the DNA is tightly wrapped around histones, and transcription factors cannot physically reach their DNA binding sites, resulting in genes stay off. So if chromatin is closed, then the genes are off. Okay, what closes chromatin is excessive zinc, but what can open it is sulforaphane from cruciferous vegetables, such as broccoli, cauliflower, kale, Brussels sprouts. Yeah, so sulforaphane is not the only thing that can open chromatin, but once it's opened, let's see here. The first food I'm going to cover is pumpkin seeds. Pumpkin seeds also have zinc, which is required for transcription factors, especially zinc finger proteins. Pumpkin seeds also has a vitamin E. Vitamin E decreases inhibitory signals on transcription factors. So what that means is that even if chromatin is opened, that even if you're trying to boost the gene transcription factors, there can be signals that can inhibit that, but the vitamin E can stop that from happening. So pumpkin seeds also have omega-6, omega-9, which affect membrane fluidity. Okay, now here's the main part. So once the chromatin is opened by sulforaphane, and the effect of pumpkin seeds in this case is improved IL-2 signaling if T cells are stimulated, because the T cells need IL-2 to be able to fight cancer cells. Tregs also rob T cells of their IL-2, so that's why it's a good thing to the pumpkin seeds to enable the transcription factors for IL-2 signaling. More efficient T cell activation because zinc is active. Zinc is also needed for IL-2 transcription complex formation. All the other things that I've been talking about in my other videos when it comes to IL-2 can't be possible alone without zinc. The other effect of pumpkin seeds is the other transcription factors that work is for reducing excessive NF-κB-driven inflammation. Also, pumpkin seeds also promote PPAR-mediated anti-inflammatory genes. So that means that genes that reduce inflammation. Also, to keep in mind that not all foods do the same thing when once their gene transcription factors enter the chromatin and interact with the DNA. And a pineapple can actually affect chromatin structure and epigenetic regulation. Bromelain in pineapple, it doesn't bind to chromatin. It actually pineapple at the chromatin state can actually reduce recruitment of histone acetyltransferases to pro-tumor and pro-treg gene promoters. So what that means is that pineapple reduces the genes for promoters that would enhance cancer tumors and tregs. So which means that pineapple at the DNA level stops the overactivation of tregs, which is used in autoimmunity. Now the reason that's important is because cancer cells, they recruit tregs, and the reason they do that is because the tregs take away IL-2. They steal it away from T cells, and what that does, how that favors cancer cells, is that the T cells are weakened because they were robbed of their IL-2. So therefore, the cancer cells are able to avoid attack by T cells. Including pineapple in my diet is one of my favorite foods, and why you should consider adding pineapple into your diet as well. So pineapples also reduce IL-6, TNF-alpha, which favors chromatin compaction at inflammatory gene loci, or the gene locations within the DNA chromatin. So the result of pineapples is a lower FOXP3, also less PDL1. That's actually a PDL1 is so basically PDL1 is something that is on tumors. So there is a PD1 expressed on T cells, and basically what the PDL1 on the tumor attacks the PD1 on the T cell, making the T cell weaker. But that's how cancer tumors actually avoid an immune attack, which is actually beyond the scope of this video. But the good news is that red cabbage can actually reduce the PDL1 expression on tumor cells via reduced inflammatory signaling, because a tumor PDL1 is often driven by NF-κB, STAT3. Red cabbage is known to reduce STAT3 because a red cabbage has anthocyanins. Red cabbage also has a sulforaphane, so red cabbage is a good thing to use to open the chromatin, because the chromatin has to be opened for the individual genes transcription factors that different foods have or boost or provide. Another good food to lower STAT3 is papaya, which has many other immense health benefits compared to other fruits. Other fruits, they don't have as the metabolic capability and benefits that papaya has. So papaya is a fruit all on its own, of its own league. That also proves that not all fruit are the same. Not to say that the other fruits aren't any good, because the other fruits do have a benefit, but they're just not as good as papaya. Okay, so back to red cabbage. So red cabbage has a sulforaphane, reduces pro-tumor cytokines, TNF-alpha, and interleukin-6. I have other videos talking about TNF-alpha, so check those out. So anthocyanins in red cabbage also lower intracellular reactive oxygen species, because PDL1 on tumors are actually driven by reactive oxygen species. So that's another way red cabbage weakens the tumor's attack on the T cell, because T cells have PD or PD1, and then the tumor has a PDL1, and the tumor attacks the cancer cell with the PDL1. So that's the end of the video. Leave your insights down below in the comments, like and subscribe, and I'll see you in the next video.